Good evening, everybody. My name is Tom Ulrich. I'm the Associate Director for Science Communications here at the Broad Institute, and it's my pleasure to welcome you all to this evening's Broad Discovery Series talk. If you're joining us for the first time, the Broad Discovery Series explores the genetic and biological roots of human health and disease, the mechanisms that govern how our cells and our bodies function, new technologies that are redefining what's possible in science, and the progress that is being made to translate new discoveries into treatments for common and rare diseases. I'm excited tonight to bring to the stage Lloyd Bod. Lloyd is a cancer immunologist, an associate member here at the Broad Institute and the Klarman Cell Observatory, a principal investigator in the Krantz Family Center for Cancer Research at Massachusetts General Hospital, and an assistant professor of medicine at Harvard Medical School. Through his research, Lloyd and his colleagues are asking how we might better harness the cancer-fighting prowess of the immune system. And tonight he's going to take us on a tour of the immune system and also give us a sneak peek into what it is that his lab has been working on. Just a couple of housekeeping notes before I bring him on stage. If you have any questions for Lloyd, I encourage you to raise your hand and flag down one of our mic runners during the Q&A portion of this evening. If you're watching us online, I'll be keeping an eye on YouTube chat. So if you type your questions in there, I'll do my best to try to work you into the discussion during the Q&A. If you're a Brody or a scientist with a deep technical question about Lloyd's research, I'm going to encourage you to please talk to him during the reception so that members of the community have an opportunity to ask their questions during the Q&A. And lastly, there is a reception. I invite you all in the room to join us in the Broad Discovery Center right outside of the auditorium, right after tonight's talk, and I encourage you to explore the museum's exhibits and interactive elements. Lloyd, over to you.
Hi everyone, can you hear me well? Thank you, Tom, for the kind introductions. It's a really good pleasure for me to be here to actually kick off the Broad Discovery series. I'm an immunologist, as Tom mentioned, actually, and I find this initiative actually critical to diffuse a bit about the science, because there is always this disconnect between what we do in the labs and actually how we diffuse the science to the broad audience. And I will bring you to a tour about immunology, which has been my passion for more than 10 years now, actually, that I realize. And so, first, what is in the name? Immunology stands for the science of the immune system, or the science of immunity. Immunity comes from the Latin immunitas, which actually comes from the freedom from obligations or privilege. So you can see it as a sort of exemption from diseases or protection from diseases. Everything is in the terminology. So today, I start to ask you questions. Why should we care about immunology? And I will involve you actually in the room. So, and you can raise your hand to answer me. How many of you know someone with an allergy? Any allergy? Almost everyone in the room, right? How many of you know someone that got the flu or got any infectious disease? Almost all the room. And I could go on if I ask you about autoimmunity. There might be less hands, but there are still many. I can do the same, and it's unfortunate about cancer. Unfortunately, many of us know someone that had cancer. The link between all of these diseases is actually that all of them involve the immune system, either in a good way or in a bad way. And why should we care about immunology? Because we need to understand how the immune system works to face these diseases and to tackle them better. Why? Because most of these diseases actually are exponentially increasing over the past years, and it will keep increasing actually in the future. You can see in orange the allergies are increasing, the autoimmune diseases, the cancer. But you can note here actually that the green line here shows the infectious diseases, and we do a pretty good job about that. Because of our way of living, we have a pretty hygienic way of life. We are pretty protected against bacterial infections or viral infections. But if you look at this timeline, it stops at 2020, and we know what happened in 2020, right? So we are not entirely protected against infectious diseases. So today, my talk will be a 5-course menu about how the immune system is composed of and how the immune system is actually playing a role in diseases, but also in normal functions of our body. So I will briefly have a historical perspective about the immune system. Then we will go together through the basics of the immune system. I will highlight how it's not only related to diseases, that the immune system have also a role in normal functions in our daily life. And we will focus, of course, on how the immune reactions to cancer occurs and is taking place. And then I will give you briefly some insights about what our lab is doing and the angle that we are taking to tackle cancer. So first, the historical perspectives. Immunology has been studied for centuries. There are so many discoveries related to immunology, many Nobel Prizes. And instead of going through each one of the discoveries, I want to guide you through main concepts related to immunology. The first concept is related to immune defense. And from the ancient Greece to the vaccines against smallpox, to Pasteur's germ theory, there is this concept of the immune system is our natural defense against bacterial or viral threats. And that's there to protect us against these threats. Then this pretty good, actually, defense system could also backfire and attack our own body. That's the emergence of this type of disease that we call autoimmune diseases. And another concept that actually took place back then is the concept of immune tolerance. How the immune system is actually educated to not attack our own body, our own self. Then more recently, over the past 50 years, I would say, and this is still ongoing, we go more into a concept of immune networks. Actually, the immune system is interconnected with our other systems, the nervous system, the endocrine system, the metabolic systems. And the immune system actually can receive signals from the environment and respond to these signals. One of the key examples here is the checkpoint molecules, the immune checkpoints, where the immune system can receive signals from the environment and that can completely change the function of the immune system. And I will talk about a little bit about the checkpoints later on because that change cancer care nowadays. So the basics of the immune systems. And the immune system is quite complex. It could be a mess because it's I would say a network, a node of multiple different pathways, different cell types, different intricacies that are melting together. But we could break it down in three main aspects. The first aspect, which is what I call behavioral immunity. This is kind of our consciousness of avoiding danger. You know, when we see a fire, we avoid the fire. And this is kind of our immune response to protect ourselves. When it's cold, right, like today, we accumulate layers actually to not get flu or to not get a cold or something like this. And also when we have a bad smell, we also have a reaction towards the bad smell. That's also a protective effect. This is kind of the first layer of immunity, which is just the behavioral immunity. The other aspects, the second aspect is more anatomical or physiological, which is really, an example is that is our skin actually represents a physical barrier that protects invaders that goes through our body. Another example also that is on this slide is the stomach that is highly acidic that can destroy everything that goes through it most of the time. So beyond the behavior, beyond the physical barrier, the immune system is then composed of cells and of product of the cells, the soluble molecules that the cells can produce. And here again, it could be divided into two main kingdoms, as the immunologists like to say. The innate versus the adaptive immunity. The adaptive is more the acquired one. The adaptive one is actually seen in the jawed vertebrates, and this is something that we have in similar to the sharks, for instance. So the innate immune system is supposed to be our first line of defense that is fast but non-specific. The adaptive immune system is composed of T cells, which contain the killer T cells that we will see later on that are the ones that kill cancer cells, and also of B cells, another lineage of this kingdom. The adaptive immunity takes longer to kick in, but can recognize specifically the target and destroy it and remember it later on. So that confers us a stronger response in the future. And you can see that there are two layers. The cellular layer on the top of the slide, where we have the cells involved in the innate immunity and the cells involved in the adaptive immunity. But then these cells produce also factors, small molecules. In the innate compartment, and we call this, I would say, non-cellular immune system as humoral. The innate humoral response contains factors such as antimicrobial peptides, which are our own antibiotics, basically. And we can directly eliminate bacteria by producing these antimicrobial peptides. On the other hand, we have also antibodies which are produced by B cells that you will see later on in a few slides, which are highly specific. And if you have to remember something from this slide, is really that the innate is non-specific, but quite fast, within 0 to 4 hours. And the adaptive immunity is highly specific. You have the concept of memory when you can remember the threat later on, but it's slower. It takes 4 to 14 days to kick in. Now let's do a little bit of philosophy. The root of immunity in general is about one simple question. How do you see yourself? Because the basic role of the immune system is to recognize what is supposed to be there, the self, or what's not supposed to be there, the non-self. And this distinction is kind of essential to understand the immune system. It's really how it works, to really make this distinction. How the immune system does that, I will give you one example. I mentioned the antibodies in the previous slides. The antibodies have this extreme ability to precisely recognize the self versus the non-self. They are small proteins which have two different portions, a recognition arm that is depicted here, and an effector function. The effector function is basically when they recognize something that is not supposed to be there, it needs to be discarded. And the effector portion of the antibody will allow the ability of other cells to pick them up and discard what is not supposed to be there. And I will highlight one of the studies that shows how specific they are. This is an example of a paper that was published in 1987, so quite old, not too old. And the researchers were looking at calcitonin. So calcitonin is an hormone that controls your calcium level in your bloodstream. And what they found, and this is the chemical structure of calcitonin that is depicted here. What they identified, they found an antibody that can recognize calcitonin. And they predicted that this antibody, that they called CT07, could recognize this portion of calcitonin. But to really test if it was that much specific, what they did, they changed the molecule. They removed this portion here, NH2, which is an amine domain. They swap it for an hydroxyl domain OH. They tested again this antibody against this molecule, but it was not able to bind at all. So this is really showing how precise and specific our antibodies. This notion of preciseness and specificity made something that we call nowadays monoclonal antibodies. So it's basically an antibody that targets a unique specific molecule. The discovery and all the studies around monoclonal antibodies obtained the Nobel Prize in 1984 by Jehan Kohler and Milstein. And it opened a whole world. It completely changed the face of biomedicine. We use antibodies nowadays in 90% of the research lab and 90% of the clinical labs to either see our cells, to treat our patients. We use antibodies every day in our daily life, as a researcher at least. So antibodies really change the face of biomedicine. So I mentioned a lot about diseases, and it's a defense mechanism. The immune system is really our defense. But then more and more that we could understand the immune system, the components of the immune system, we could also realize that the immune system could play other roles beyond just diseases. Here, I don't know if you can see, This is a review that was published in 2018, where the authors basically summarize what are the non-disease functions of the immune system, here depicted in blue. And we could find in the literature that the immune system can regulate basic functions, such as how we burn fat, for example. They regulate thermogenesis in the white adipose tissues, or the brown adipose tissues as well. They regulate the heart functionality. They regulate how we generate our muscles, how we regenerate our muscles. The immune system also controls how the motility of our gut. And these are functions, basic functions that are routinely involving the immune system in their actions. And for me, one of the mind-blowing examples, and I will show you one of the paper after the slides, is the recognition that the immune system can even control our own behavior and our social behavior. You remember at the beginning of a few slides, I mentioned that our consciousness provides a behavioral immunity. But now it's more the retroaction of the immune system on our behavior. And this is a growing literature where they actually recognize that this is happening most of the times. So one example is this paper published in 2016 in the journal Nature, where the authors interrogated the role of interferon gamma. Interferon gamma is a small molecule produced by the immune system, mostly T cells, or the killer T cells, that regulates viral infections or that plays a huge role in cancer as well. And to test the role of interferon gamma on the social behavior, they use a pretty interesting model. It's called the 3-chamber sociability assay. In this assay, they have a box. They put a mouse into the box. The box has 3 chambers that are interconnected with these openings that you see here. On the left chamber and the right chamber, you have separate cages within the box. And at the beginning, they let the mouse go and discover the box itself and move between the right and the left chambers. After a while, they would introduce either another mouse in the left chamber or an object in the right chamber, for instance. And you can directly understand the system. It's pretty simple. If the mouse is pretty social, it should spend more time with the other mouse, right? And if there is no social aspect, if the mouse doesn't really care, the mouse would spend as much time in any chamber, right? And mice are like humans. They are quite social, actually. They spend twice much time with the other mouse compared to the object in a normal situation. However, without interferon gamma, that's the molecule that they investigated, then these mice lost completely their social behavior, which shows that one single molecule produced by the immune system can control the whole social behavior of these animals. So beyond just diseases, we have also this interconnection between these 2 different systems. And the immune systems will, I'm pretty sure, will teach us many more about our social behaviors and more about our consciousness as well. So now let's dive into the role of the immune reactions to cancer. So the title here is about the cancer immunity cycle that has been recognized for the past 50 years. I have to say that these mechanisms is actually true, which is for infectious diseases, for allergy, for autoimmunity. This will be the same kind of cycle. And the cycle is composed of 4 different steps. The first step is the fact that the immune system is there patrolling throughout our body to find what is not supposed to be there, right? So the first step is really to identify cancer cells that would not look quite right? There would be subtle differences that would make the cancer cells becoming different than the normal cells. And these immune cells would then recognize the ID card of the tumor cells, of the cancer cells. This is the greatest, prettiest picture of tumor cells. Then once these immune cells would would get the ID card of these cancer cells, they would then migrate to an immune organ, such as the lymph nodes or the spleen. And they would present this ID card to the immune cells resident there, either the T cells or the B cells. In that case, in the case of cancer, they really want to show this ID card to the killer T cells, right? This step, the second step of the cycle, is called the immune priming. We call a primed immune cells when the immune cells is conscious that there is a threat to attack. Once the cells are primed, then they can migrate within the tumor tissue and do their job of destroying cancer cells, right? And I give you one example. So this is a microscopy picture of immune cells that are here in green, surrounding a tumor cell that I don't know if you can see the color. It's supposed to be purple, and that are doing their job of destroying the cancer cells. And this is how, in theory, it should happen. And you will tell me, so Lloyd, if the immune system was so great, why cancer is still a thing, right? And you're right. There is a mechanism that is a sort of double-edged sword, where by being so effective of recognizing and destroying cancer cells, the immune system also provokes something called immunological sculpting. This concept has three phases. The first one is the job is done quite well. The first phase is the elimination phase. The immune system recognizes and destroys most of the cancer cells. However, in the subsequent stage, sometimes they can be outnumbered. And then they could actually maximize their effect. They destroy most of the cancer cells, but there are still some that are remaining. This is called the equilibrium phase. And in that phase, it's really kind of keeping a wild animal into a cage. So you can control the growth, you can contain the wild animal, but it's still there, right? And what is happening during this phase, the elimination phase, is that some cancer cells can actually learn how to overcome the immune attacks and outgrow and develop into real tumors that are on the right of the slides, which is called the escape phase or the evasion phase of the immune system. And you can relate it to a sort of Darwin evolution concept, where the cancer cells would evolve to adapt to the immune attacks to actually survive. And this is really similar mechanisms that are occurring here. Then, in that case here, why then the immune system wouldn't be able to still attack these cells? What are the mechanisms of evasion that makes the tumor so resistant to the attacks? And there are different strategies that the cancer cells have actually to evade the immune system. I will highlight two of them here today. One is that the cancer cells can become invisible, completely invisible. They can hide from the immune system. How do they do that? I don't know if there are some Game of Thrones fans in the room. They can actually build a wall surrounding them. They can corrupt the cells around them to make them build a wall surrounding the tumor tissue. And this is not fantasy. I can show you an actual microscopy example. This is a lung tumor. In blue in the middle, you can see the cancer cells. In red here, you have the fibers of fibronectin that are really building this wall around the cancer cells. And the immune cells in green here, you can see them, they are stuck outside of the tumor tissue. They just cannot get there. In that case, the tumor is completely invisible and the immune system cannot do anything. So that's one of the evasion mechanisms that is quite striking on this image here. Another strategy is, even though the immune cells can get there, cancer cells can become highly resistant. And I will highlight there are many resistance mechanisms, but one of the key resistant mechanisms is depicted in this cartoon here. Remember this slide when I showed these cancer cells surrounded by immune cells that tried to destroy it? What could happen is cancer cells can also hijack one of the normal functions of the immune systems called hijacking the checkpoint molecules. So in the first panel of the cartoon, the T cells in blue here, which I don't know why they are archers in that case, have the ID card of the cancer cells in their hands, and they are supposed to attack them. However, the cancer cells can fight back by using PD-L1, which is the baseball bat, and target PD-1 on the surface, on the head of the T cells. This would completely dismantle T cells. They would be dysfunctional, and then the tumor can then evade the immune system. Again, here, instead of a cartoon, I wanted to show you what is actually, when we look at it in situ, when we look at a human tumor, by giving an example of colorectal cancer. In pink here are the cancer cells. And in the light blue, you can see the immune cells. The first thing that you see in this microscopy image is that, again, you can have a sense of the wall, that the immune cells, right, seems to be stuck outside of the tumor tissue, right? So you have also so none of them are actually separated. They can all converge and be in play in the same time. If we zoom on one of the regions, for example, here in this region, when we can see that there are some immune cells that could get there, right? What we see, I don't know if you can actually see that nicely here, it's all of these cells in orange or red, as you can see, express PD-L1. So the bat is there. Even though they made it there, they could get through the tumor tissue. They still are completely dysfunctional because the PD-L1 is expressed highly on the surface of the cancer cells, and that dismantles their function. So this is an active mechanism of resistance by the cancer cells that really can avoid the immune destruction. But what can we do? If there are so many different strategies of invisibility, of resistance, how can we actually boost the immune response to the context of cancer? And what are the strategies that we can do? In clinic nowadays, there are 3 main therapies. So therapies that affect the immune system are called immunotherapies. So I will use this terminology for the rest of my talk. So cancer immunotherapies have 3 kinds. One is modulating these checkpoints, the signals that actually inhibit the immune system. One strategy is if the immune cells are outnumbered, then let's just put more. So it's a strategy of actually overcoming the low amount of immune cells in the tumor tissue. And the last strategy is something that has been around from the beginning of the world of immunology, is making cancer vaccines. So actually preparing the immune system to recognize better cancer cells, to destroy them even before they would outgrow and become a real tumor. It's really trying to stop a fire before it spreads, a small fire before it spreads. I will give you one example of each strategies. The first one is immune checkpoint blockade therapy, where in this cartoon again here, you can see pretty simply that the strategy is just if the cancer cells can use a bat against the T cells, the killer T cells, let's just put a helmet on these cells, and then they will be protected. And you know what? That works. That actually works. By adding this helmet, the PD-1 checkpoint blockade, we would actually protect the T cells from being inhibited by cancer cells and allow them to work better. And then they can again fight cancer. This strategy got the most recent Immunology Nobel Prize in 2018 by Jim Allison and Tatsuko Honjo. But I also want to highlight people that were also involved in the discoveries, Liping Chen in Yale, but also 2 Brodies that are also part of the community, Gordon Freeman and Arlene Sharp. Because these type of therapies were really game changer in cancer care. Some cancers that were completely untreatable with chemotherapy, the standard of care, were now actually had some hope because of these immune checkpoint blockade therapies. The second aspect is also quite simple in the reasoning, right? It's just there is not enough immune cells, let's just put more. One of the strategies actually is highly clever, is using the best of both worlds. We know there are these killer T cells that are highly efficient of recognizing, remembering, and killing cancer cells. But we know also that antibodies have this highly precise and specific way of recognizing what is not supposed to be there, right? So let's just combine them. Let's just put an antibody on the surface of T cells. This is called CAR T cell therapies. And the principle is that we would take T cells from patients. We would genetically add the recognition portion of the antibody on the surface of these T cells, the CAR. We would multiply them in vitro and then put them back to the patient. And that would actually enhance the recognition of cancer cells and also enhance the killing aspects. These CAR T-cell therapy completely cured some cancers. Some lymphoma were completely cured thanks to the CAR T-cell therapies, for example. The other strategies is still controversial nowadays, I have to say. It has been around for decades and decades and decades, but they were not pretty effective 50 years ago. The cancer vaccines, I took this magazine review that was published in 2004, so 20 years ago, and back then they were already questioning if it is a hype or a hope. And I have to say, for the past 20 years, the results were not quite right. However, since 5 years, since 2020, everything changed. And you will understand why. There are so many different literatures that are like reviving the field of cancer vaccines because of specific new strategies, new, I would say, tools to improve cancer vaccines. Highlighted in green or in yellow some names, which are basically the names of the researchers behind Biotechne and Moderna. We all know that with COVID vaccines, the new methodology behind vaccines could actually bring more hope into cancer vaccines right now. And there are some recent clinical trials that show that some cancer vaccines are highly effective in cancers that were completely refractory to any therapies, such as pancreatic cancer, for example, here. Because of all of these years of strategies, understanding better how the immune system works, understanding the cancer immunity cycle, understanding the different components of the immune system, it finally got a place as one of the 5 pillars of cancer treatment and cancer care. Originally, there was only surgery that was feasible for those patients. Then there was the emergence of radiotherapy or chemotherapy that is still heavily used, these two. Then became the world of targeted therapies and immunotherapies. Despite the fact that immunotherapies could change cancer care nowadays, it's still quite limited, I have to say. We are still at the beginning of the revolution regarding immunotherapies. And I will give one example here. This is a Kaplan-Meier curve, so this is the survival curve. For the people that are not really used to these curves, this is basically showing the survival of the patients over time between 3 different groups. Either patient that got 2 different types of immunotherapy, nivolumab or ipilimumab, which are just different types of helmets, basically, the green and the blue. And the red is basically adding the 2 helmets on top of each other. Because the rationale again here was maybe we can improve the protection by adding 2 helmets at the same time. And you can see that it works, actually. By adding only ipilimumab, we got 27, so 31 patient out of 3 actually responded pretty well and survived well to the therapies. By adding this helmet in blue, it's actually more effective, 1 patient out of 2. But combining both gave rise to almost 60% of the patients that would respond well and had survival benefit with immunotherapies. But these numbers also mean that 40% in that case would fail to respond to immunotherapies. And we have to understand also that immunotherapies, the principle is quite simple. It's boosting our own immune defense, right? Our own defense. So why 40% of the patients would not respond well? And this is by the combination therapy, but here, half of the patients do not respond at all, and here, 70% of the patients do not respond at all. It's always nice to look at the numbers the other way around. And the goal is really to understand why immunotherapies are not applicable to everybody, and also what can we do to improve therapeutic efficacy. And right now, most of the field has been focusing on the helmets. So basically trying to make fancier helmets, heavier helmets, combining different helmets, but the numbers are not improving just by trying to put more helmets on the surface of killer T cells. And the field is more recognizing that the immune system is more complex, of course, that we have seen earlier, but is not only composed of killer T cells and cancer cells, the killer and the target. There is more complexities and more different flavors of immune cells. Can we harness these other immune cells, actually? Here, I switch gears. I showed you lung cancer, then colorectal cancer. Here it's an image of melanoma, skin cancer, a type of skin cancer. The cancer cells are depicted here in green and blue. And in gray are all the other cells in the environment. And here on the same image, then I highlighted these other cells and to show you what they are. You can see that the killer T cells are these cells in light blue. You have one here, you have one here. But you can see also a bunch of other cells, right? B cells in red, depicted here, the one producing antibodies. Helper T cells, another type of T cells in orange, here, here, and here. And also macrophages. Macrophages are the immune cells that are from the innate part of immunity that just eats everything. So they are super efficient there when you have a destroyed cancer cell to actually clear the place and discard actually the dead cancer cells. So it's more complex. And the question around that is, can we try to change the environment to provide the killer T cells a more supportive environment and not trying to make them work harder, faster, better? It's also a philosophical concept about how we would treat our trainees nowadays at the same time. So I will give you now some insights about the lab, because this is kind of the core of our research, is trying to find a new angle to tackle cancer. The angle we chose is to leverage B cells, the other immune cells from the adaptive kingdom that are producing these antibodies. And these are these cells. At the beginning, they look like these cute cells that could be useless. And then when they get angry, they produce all of these different antibodies that are highly specific. Why should we care about B cells? It's because they are everywhere and in all the cancers as possible. On this plot, this figure, you have each column represents one different cancer types. So you have 29 different cancer types. In gray, each stack are different immune cells. In purple, I highlighted the B cells, and you can see that they are present everywhere in each one of different cancer cells. However, the way we look at this is nice, this is correlative, they are there. But how can we predict their function in these different cancers? A simple way is to relate the abundance of these cells and the outcome of the patient. To look into the more a patient has B cells, how does that change the survival of the patient? And this is a summary here, because this is quite controversial in the context of B cells. In some cancer types, such as breast, lung, or skin cancer, the more a patient has B cells, the better it is for the survival. They survive much better. In some cancers, there is just no effect. That's the case of brain, colon, or kidney cancer. You can have like more or less B cells. That doesn't change at all the survival of the patients. And in some other cancers, such as adenoid or pancreatic, it's actually the other way around. The more you have B cells, the worse it is for your prognosis. So this controversy could be explained by the different flavors of B cells that are found in these cancers. Maybe that's not the same type of B cells in that tumor, then that tumor, then that tumor. And that might explain why you have these discrepancies. There might be bad versus good B cells. And that's kind of the core of the lab, to identify these different flavors of B cells and then test them. Another philosophical question for you. To be or not to be? This is the core question of the lab, and I hope we are B. The lab is really trying to understand the mosaic of the flavors of B cells within cancers, in all the different cancers. That can help us to identify actually the good B cells and the bad B cells. The goal would be really to find the one that can promote tumor control, to keep the tumors on check, and the one that can promote tumor growth. Again, are they good versus bad B cells, and how can we actually define them? What do we do with this information? When we have identified bad or good B cells, the goal would be to then target them, to find checkpoints like have been found for T cells in B cells. And that's the second big aim of the lab, big goal of the lab. And we were fortunate to have dived into B cells that much in melanoma, colorectal cancer, lung cancer, or breast cancer, that we could find some molecules that could promote good B cells and could inhibit bad B cells. And we are continuously working about these molecules nowadays. I will give you one example of one of them that we recently discovered that is called TIM1. That is a molecule that is specific. It's an equivalent of the PD-1 on T cells that I've shown earlier, where when this molecule is blocked on B cells, then the tumor can barely grow. This could offer an opportunity to tackle this other angle of the immune system by harnessing and exploiting B cell functionality. So this could lead to more promises in cancer, because instead of adding helmets, we would add one helmet and maybe also boosting another immune cells to combat cancer better. Beyond all of this, what we find when we analyze so many different flavors of B cells would be also biomarkers. And this is really something also that we are investigating in the lab. One of the key goals of these types of analysis is to identify cancer fingerprints. Imagine if we can predict if an individual will develop cancer. Imagine if we can actually predict if a patient will respond or not to immunotherapy. That would be key component to help for the diagnosis or actually for the management of these patients. And to do this, we study antibodies that are produced by B cells. And we recently collaborated with a team in San Francisco at UCSF, where what we did is we profiled antibody in over 700 individuals. These individuals were either lung cancer patients at risk of developing lung cancer but asymptomatic so far, and healthy individuals. Then we developed an AI model that could accurately distinguish if a patient had cancer or was healthy. And we could, with this tool, efficiently be able to improve potentially the diagnosis of this patient with lung cancer. And we hope to expand this type of analysis to any kind of cancer. The first stage was identifying cancer versus not cancer, so what's supposed to be there or what's not supposed to be there, the self and the non-self. But later on, the goal would be really to identify the ones that would respond or not respond to immunotherapy, to chemotherapy, to radiotherapy, and so on. That could really help the management of cancer patients. So this is the core of the lab. I'm good, I'm not too late. And as conclusive notes, I would say that the immune system are like ants. This is how I see the immune system to me. I see as a highly, highly organized melting pot of different ants that serve the same goal, that works together, that could be dangerous sometimes, right? But at the same time, that would have these intricacies that makes them to overcome many different challenges. And there are still challenges in cancer immunotherapies. I mentioned some of them earlier, that not every patient is actually responding to immunotherapies. And I wanted to finish on the key future challenges for cancer immunotherapies. Obviously, we need to improve the response. We need to find why the patient do not respond and improve the response. This also implicates that we need to understand the resistance mechanisms. I give you two examples, the invisibility or just the basic resistance with checkpoint. But how can we alleviate this resistance? That's something that we really need to understand better. Most of the field right now is focusing on the resistance. They are trying to add helmets to make the cells work better, faster, and be more aggressive. But you know, the immune system is also dangerous, as I mentioned, like the ants. Because if you boost it too much, it could go rogue and provoke toxicities and even autoimmune reactions, right? So at the same time that we try to improve response, to alleviate resistance, we also need to avoid toxicity. As an example is in numbers, Mass General Hospital and Brigham and Women's Hospital treat over 4,000 patients per year with immunotherapy. Out of these 4,000 patients, if they were treated with a single agent, only one single helmet, for example, 7 out of 10 patients would develop toxicity, but fortunately, only 1 out of 10 would develop severe toxicity. However, if you combine immunotherapies, the 2 helmets that provide the most response, the 60% response, then 9 out of 10 patients would develop toxicities, which is a huge number, and half of them would develop severe toxicity. So really, the goal wouldn't be to add helmets on these cells, would be mainly to find other angles to improve the response, right? Avoiding toxicities. So we recently joined the forces of a pioneer in the field of toxicities, Chloe Villani, that is depicted here, with Will Wang and David Ting. And we formed this new project called PROTECT, which stands for Promoting Response to Oncoimmunologic Therapies while Eradicating Toxicities. The goal of this project now, we actually bridged our forces to understand the components of the resistance, the components of the response, but also the components of what makes it toxic to alleviate this toxicity, these immune reactions. So wish us luck for that. So overall, these are my last slides. To learn more about immunology, which is really an amazing field to learn more about, you can go onto the different societies, so the American Societies of Immunologists or the International Society of Immunologists. Some of the cartoons that I have shown, I took them from UCIR, which is a great resource to explain immunotherapies to the lay audience. And every April 29th is the International Immunology Day. And there are many events across town and in the world, so please look out for that. And then if you have kids or teenagers, I highly recommend this book that exists in many languages about the immune system. And that's kind of a fantastic resource, and you have the QR code here to download it. With this, I really want to thank you for your attention. Here is my lab that is between the Broad and Mass General Hospital. And then we recently won the Lab Olympics last summer. And what we won because of what? Because we had the best slogan, which was, "Let it be." One last slide about some recent events. You know, in research or in clinic, cancer care and all of these therapies and all of these studies wouldn't exist, wouldn't actually be happening without the infrastructure, and especially the administrative infrastructure. So I want to take the opportunity today also to acknowledge them, both at MGB, Harvard, or also the Broads, that without them, I wouldn't be standing here today, and the advancement in the field wouldn't have happened at the same time. Thank you for your attention. That was a pleasure to be with you tonight.
Thank you so much, Lloyd. Now, if you had told me ahead of time that April 29th was International Immunology Day, we would have scheduled the talk for then.
Mm-hmm. Sure.
If you have any questions, the mics are yours. We have a couple of mic runners here who are here to hand things out. But before we actually dive into the audience questions, I just wanted to ask you a question really quickly, Lloyd. Can you just describe real quickly your career path? How did you get interested in immunology in cancer, and how did you come to be at the Broad and at MGH?
Yeah. So it's an interesting path. When I was a teenager, I grew up in Martinique. That's a small French island in the Caribbean. You could tell from my first sentence that I was French, right? My accent, I cannot hide it that well. And I grew up in Martinique, and I was always interested by surgery, actually. I went into a MD PhD program in Paris, and I did many, many surgeries. Then I remember that class when they were teaching immunology, and that completely snapped my brain. I was so fascinated about all the different possibilities and complicities of immunology, so I decided to move on and do a PhD in immunology and so on and so on. And then my uncle developed melanoma, advanced metastatic melanoma, and he got, in 2012, he got actually in a complete remission with anti-CTLA-4, anti-PD-1, so the two helmets that I've shown earlier, and that actually prolonged his life for 10 more years. And that also is why I decided to focus on cancer immunology and moved here to develop my program.
And to make better helmets.
Yeah, exactly.
I think we had a question right down the front here.
Yeah,
Yes.
so my question is about the immune system and what happens to it during pregnancy?
Oh, great question. So remember about the concepts, the defense, the tolerance. The pregnancy is such an example because the fetus is actually quite not exactly self. It should be the mother self, right? But it's half of the father self. So the immune system is actually trying to attack it because it recognizes it as something that is not supposed to be there, which is not self. So again here, the immune system learns how to tolerate the fetus and allow the fetus to grow. And that mechanism is really about, I use a lot of terms that are used in the society like education. The immune system then gets educated to tolerate the fetus and let them grow. That's a great question.
Question over here.
Yes.
Can you hear me? Yeah.
Yes. I do.
So you talked about hygiene and innate immunity.
Yes.
Right? So I have a two-part question. One is too much hygiene a bad thing? And number two, isn't cancer a last 50-year environmental, thingy that's come up now, and we're all trying to work on it and figure out what's going on?
Yeah.
And it wasn't there many years ago.
Yeah. So for the first part of your question, it's actually a good point. There was in the 1970s the hygiene theory, meaning that the more we are protected against infections, the more we are weakened, and the more the immune system doesn't know what to protect ourselves from, so it starts to attacking ourselves. And this is the hygiene theory, which could be the case in some cases. And there are studies, unfortunately, in the statistics that do not show that the hygiene theory stands. But this is theories that are out there. In the case of cancer, with the numbers increasing over the past years and so on, since the 1980 to now, so it's not entirely fair to say that they increase. It's also that we diagnose them much better. We have the technologies nowadays to diagnose so efficiently cancer at the single cell of cancer that a patient has, or to diagnose allergy very quickly. So the tools change. So we see more of them, maybe also because we have the tools to see them more often. But what you said is also right, that the way of life, the new environmental changes, the pollution, the diet, and so on, are also factors that provoke cancer more efficiently. One of the saddest part of it is that usually cancer is seen in elderly populations, but nowadays we see more into young people. And these young onset cancers are now a question about why do they rise so fast, and actually they are also more aggressive in the young populations. So you're totally right. These also are questions about the way of life. Thank you.
Actually, while the mic is making its way, so you mentioned T cells a lot.
Yeah.
You mentioned B cells a lot.
Yeah.
Are there other cells within the immune system that also play a role in cancer surveillance, in attacking cancers?
Yeah, totally. So I mentioned a lot T cells and B cells because at the end, these are the only cells that can carry this memory. And this feature of the immune system to remember the threats is kind of what we really need to go after to prevent the occurrence of cancer later on. But you're totally right. There are other cells in the innate kingdom of the immune system that are highly effective to I mentioned the macrophages that's really helpful to actually clear out cancer cells. But you have also a cell type that has a name pretty cool, which is natural killer cells. And their function is to kill. Their basic functions, they will not check the specificities, they will not have the memory aspects. They will just have an on and off role of killing and not killing. And this has play also an important role in cancer. Yeah.
Question over here.
Hello.
Yes.
It was really interesting talk.
Thank you.
I want to ask about what technologies are currently innovative to understand better the relationship between all the types of immune cells, and also if you think like, because your lab like transomics, that you also do like research on that, like how do you approach?
Yeah. So thank you for that question. Some of the key technologies were actually from the Broad, and they revolutionized the world of research nowadays again. Monoclonal antibodies were the first phase of revolution, and the genomics field were also the second phase of revolution. Now we can understand at a high depth each single genes, each single proteins that are produced by each single cells of the body, which is too many data. But with this, if we have a focused question, then we can actually leverage these technologies to understand better the relationship between cells, and also their different flavors, their continuity, so the dynamic over time, before therapy, after therapy. And this is something that we leverage here, the genomics aspect a lot in the lab, at the Broad, and at MGH.
Thank you.
Thank you.
So I could just repeat the question so
am.
the people watching online.
Yeah, the question is related to how T cells and what T cells recognize, if all T cells recognize the same thing, or you have different diverse T cells that recognize different things, and then how they grow. And then you touch on also a unique feature of T cells and B cells, which is that when they know what they should recognize, and they can recognize many different things, when each one has a specific target in mind, they can actually multiply. So we call that clonal expansion. So they expand toward a specific, targeting a specific unique molecule. And you can have multiple different clones, so they recognize multiple different antigens that we call, which are the different targets. And that is happening for T cells and B cells, yes.
I'm trying to remember how many, like, I know there's a number out there, like a theoretical number of how many different targets your immune system is able to handle. It's like 10 to the...
to the power of 18.
Yeah.
It's really trillions and trillions and trillions of targets. The diversity, we call it the repertoire of the immune system, is so diverse. And that evolved actually across the normal evolution of the human being to be able to recognize so many different threats. So the diversity is also a function of our protection to recognize any flavors of bacteria, viruses, cancers that we can encounter. Yeah, good question.
Question up there.
Thank you.
Yes.
I have a slightly specific question. For the CAR T therapy, how do you design the chimeric antigen receptor? Like, what's the in vitro strategy to make sure it's specific to the tumor?
Yeah, so first we have to have in mind a target. We have to have a sense of what the cancer looks like, right? So CAR T cell therapy, where the proof of concept was established with lymphoma. So lymphoma is actually a cancer of our own immune cells. In the B cell lymphoma, that's our own B cells that would expand in an anarchic way and would really become their own tumor, right? But there is something in common, is that they are B cells. All of them are B cells. So they have proteins of B cells on their surface, even if they became cancer cells, right? So we could arm, that's how we call it actually, the CAR T, the T cells, with only the recognition function of the antibody that we took from a monoclonal antibody, mount it on the surface of the T cells against one of the B cell proteins. And then they would kill every B cells, not only the cancer cells, actually. So that's a great point, because now it's really difficult to make the distinction between what is the ID of an exact cancer cells or the ID of just a normal cells. Can we make the distinction between normal pancreatic cells or pancreatic cancer cells? This is really tricky questions now. With CAR T cells, it's still limited to make this distinction, but the field is moving pretty fast toward identifying the subtleties between the two.
Thank you.
Good question.
One more question over here.
Yes.
When you're developing immunotherapies, have you found much commonality across patients in the antigens you're trying to develop antibodies against?
Yeah.
Or is it just very individualized? And even within a single patient, do you find different clones that have different antigens that you have? How much customization do you have to do, or are there patterns that you found that you?
Yeah, no, that's a great question. The dream would be to have an off-the-shelf therapy that could work for every patient, right? The helmet is kind of this off-the-shelf therapy. Then we would be agnostic of the antigens, of the type of cancers and so on. We would just give a helmet to the T cells, and they would recognize whatever they recognize and then kill better, right? However, it's still failing a lot. So now the field is looking more into distinguishing at an individual level what are personalized to that patient, to that cancer types. And that's more related to the CAR T therapy, for example, that will evolve, or also the cancer vaccines. And more that's more related to the cancer vaccine phase. For immunotherapy globally, it's mostly trying just to boost the response without having every specificity patient to patient, actually. Thank you for the question.
So, last question.
Yeah.
5, 10 years from now,
Yeah.
like, how do you see this story of cancer in the immune system unfolding?
I think more and more we will hopefully have the strategy of promoting the other immune cells, involving the network, the interconnection between the system. Not only trying to put a helmet stronger, but really trying to understand better when the tumor evaded the immune system, which cells were involved in the invasion, where does that happen, what is happening. And by understanding, unfolding all of this with the new technologies, I believe that in 5, 10 years from now, there will be more combination therapies, but instead of combining on the same cell type, we'll be combining across different cell types and different contextures. And hopefully, there are many promises, but the cancer vaccines might be also effective in 5 to 10 years. Fingers crossed.
All right, so Immunology Day, 5 years from now, we're getting you back on stage. And we're going to see if you're right.
Okay, sounds good.
Thank you so much, Lloyd.
Thank you, Tom. Thank you so much. Thank you.
And thank you also to all of you in the room and online who have been watching us tonight. We will let you know when our next Broad Discovery series will be. It's probably going to be sometime this spring. I hope you all join us then again. And with that, have a great night.